Hello and welcome back to biology. So what I'm going to do now is the functions of organelles. So as you recall, I looked at two different eukaryotic cells, and that was the first animal cells in biology 1, and then plant cells in biology 2. So what I'm going to do is obviously I drew a lot of different things, and now what I'd like to do is on the left hand side provide a description, and then just draw a little picture on the right hand side, just to give a bit more detail, but the pictures not be hugely detailed, just to reiterate what I've gone through before. So yeah, so we're going to start now with the plasma membrane. So maybe I will just draw a box. This could be the plasma membrane. So what can we say about the plasma membrane? Well, it's also called the cell surface membrane, and it's really a key component of the cell, and has much to do with transport. Phospholipids, very important when it comes to that. Yeah, let's think of a definition. And it's basically it's on the surface of animal cells. Yeah, I think I might just actually put it just below. And inside the cell, or just on the edge of both plant cells and eukaryotic cells. Okay. So made out of lipids and proteins. Okay. So that can be seen as a description. So what I might do actually is, just to vary things a little, I might just change the colour to blue, and I can say that blue is going to basically indicate the function. So yeah, seems like a fairly good idea. Okay, so I've got the blue now. Hopefully, yeah, it looks the same. Good. Right, so what it does, it regulates movement in and out of the cell. Okay, it has receptor molecules, which to as big gap. Oh well. Now to respond to chemicals. Yep. So very briefly, I'm just going to be very brief with this. It's literally you know, here's the plasma membrane. And and then you have, it's just, I'll just and then we've got the cytoplasm right here. Yeah. Alright, so I think that will do it for the plasma membrane. So we use this colour. Okay. So there's a plasma membrane. I think I might actually get to the end of the page on one of these ones. Alright, so the next one we're dealing with. Cell wall. Okay. Okay. Very rigid structure. So it wouldn't be cool, it's not amorphous. I wonder if amorphous is actually a word. Might be. It's interesting that there's morphous and there's morphology. Okay. And it surrounds plant cells, of course, as we saw in the diagram in biology 2. Okay. This is mainly of cellulose, yeah. And how did I do that? I just, yeah. Plant cells. And once again, well, perhaps I should have. Let's try. I can do the plasma membrane again. Actually, what I'll do is I'll just do the plasma membrane. So there's the plasma membrane. Again, we're going to have the cytoplasm down here. And then we'll have this this layer to be the cell wall. So we'll just so yeah, just like this. Cell wall. Cell surface membrane. Let's put plasma in there. And once again, cytoplasm, right. Okay. So you're cool now. Oh yeah. There's the end of that one. Cool. Oh. Alright. nucleus. So the nucleus is good for eukaryotic cells. We'll go back to red again. Nucleus is always a big one, isn't it? It's a large organelle. Got an important name. Yeah. Okay. Okay, and as you saw in the pictures, surrounded by nuclear envelope. I'm actually going to talk a little bit more because there's actually chromatin. Outside it's outside the nucleus, but inside the nucleus. What was I going to put here? Oh yeah, let's speak about the envelope first. Okay, there's a double membrane. So there's quite a lot of membranes in this glossary of organelles. And like I just said, and just a further note about the nuclear envelope, it's porous. Okay. So contains chromatin, which I did in some of the features. I tried to make it look like, there's little dots and stuff. And the nucleolus structure. I've actually found I quite like working with a pencil with a fairly. It seems to have a good, because this text is much more readable than normal. I like using this pen there. I like to write with a ballpoint pen really. What else can I say? Okay, so we're going to move into functionality. Just remind blue is functionality and red is description. Okay. So chromatin. Made from proteins. DNA. And obviously, something like DNA, you have to speak around a lot more, but not now. But you can control some activities. Okay. Good. So these pores, in the this is not in the nuclear envelope. They're just pores exist. There are gaps around the nuclear envelope. Okay. So they allow RNA just between. Cool. And the function of the nucleolus um, um, makes ribosomes, which of course you've seen in the picture as well. Um, so picture time. Wow, this can be an interesting one. I think I'm going to try the envelope first. I'm going to leave some little gaps here. I'm going to make this much smaller actually. Just going to draw the outline here. Make that a little bigger, just to here. Of course, it may be a little bit irregular like this anyway, so not too bad. Okay, so now how about the nucleolus, making all those ribosomes? We'll make it a big blob. Let's make it a little bit funky. Yeah, there you go. And what about the chromatin? I just, okay. So yeah, I think with these things it's always bad just to, just try and keep it simple. So I suppose I just write. So this is a pore, showing the porous structure, and of course the nucleolus. Great. Okay. Wow. I would have thought 4,000 pixels would be enough, but maybe not. Alright. Let's move on. Now lysosome. Now you recall that we didn't draw these in the plant cell. We did draw them in the animal cell. So, something to note. Okay. So we have a round organelle. Surrounded once again by a membrane. And it's almost a little bit unfortunate when I think about it actually that I haven't drawn it like that, but I probably, to be honest, I'm probably gonna draw these things a lot of times. Yeah. Generally, something hasn't got clear internal structure. It's probably because there's some stuff going on at smaller size. Functionality of this. Yeah, I mean, I suppose. Yeah, that is a bit of a problem. I haven't drawn it. But I mean, can you actually do that? Have a paintbrush with a membrane on it? I wonder. You can't really. Yeah, I do wonder about it a little bit. Yeah, I mean, if you start with this, I suppose in the animal cell I did do it better, but anyway. I mean, just thinking about different ways of drawing. I should really look at the um, I should look at the the manual, the help document for paint at some point, because it might be a bit easier to do that. And yeah, I don't know why I thought of it from the lysosome, but it just seems a bit seems kind of relevant. Enzymes. Yeah, for some reason I think well, I suppose I can say I think of enzymes and catalysts as kind of similar. But I mean, that's the thing about biology. You don't really want to say too much. But then again, sometimes if you have a good idea and you can link this stuff together, it's not really too bad. But I think you have to know about the different. If you're trying to tease stuff, try not to do that too much. Yeah, that's just mostly it, really. All right. So lysosomes contain digestive enzymes. The membrane keeps the enzyme separate from the cytoplasm. Okay. You see, this is not. You see, yeah. So enzymes used to digest invading cells. So yeah, that was not really. Oh, wasn't necessarily a bad way to put it. Cool. Well, it's not the most interesting picture. Because here I can draw an outline for it. I'll just put in a load of dots. And a fairly grey sort of colour. This is going to go over it though. So I probably should have done the dots on top. It doesn't matter that much. It's only a few dots. So yeah, no real clear goings on. It's because these things are pretty complicated. Start looking at them through smaller units, I suppose. So that is, in a way, it looks like a big full stop. So that is where I'm going to leave it.